What is disease? Disease is a state of dysfunction when the body is not working as it should or as efficiently. Disease can come about through environmental exposures and toxicities, genetic or heritable conditions. You need 3 requirements for disease. All 3 must be met simultaneously. The first thing is you must have a pathogen. Remember that most microorganisms are good. The second is you must have a conducive environment, meaning there has to be a mechanism for that pathogen to get into the cells that it's likely to infect. So a means of transmission. And third, you must be a susceptible host. And remember, we have our innate system there, which largely makes this a hurdle to overcome. Our immune system does its job so efficiently that we rarely notice it is at work. So our body has different ways to protect us against disease-causing invaders. We define the body's ability to ward off disease as immunity. Why don't we get sick more often? If bacteria are ubiquitous and they are around 24/7, 365 days out of the year, why is it that we only seem to get sick maybe once or twice a year? That's largely due to our immune response and the ability for it to remember and therefore protect against repeat exposure. We begin by talking about the innate immune system. Be able to recognize the 4 major functions of this system. First and foremost, it's to prevent entry to the host. Now, assuming that something gets in, the innate system will then try to trap, contain, or limit the spread of that pathogen. Function 3 is to kill or clear and remove the pathogen from the host body. And then 4 occurs when the innate system recognizes it's overwhelmed and it starts to coordinate with the adaptive immune response. With respect to preventing entry, there's a number of mechanisms in place, a number of anatomical structures able to help minimize entry of a microbe or pathogen into the human host. For one, skin is composed of tightly packed cells, many layers thick. The top layer itself is dead and dry, and so this is not an environment usually conducive to osmosis and diffusion. It's also very difficult for a microorganism to burrow their way in through that. Wherever we don't have skin, our orifices or openings, there's a general theme of slippery when wet. And the idea here is our earwax, our nasal drippings, our saliva, it's meant to wash away or dilute numbers of incoming pathogens. An interesting thing I'd like to point out is the enzyme lysozyme. Recognize this enzyme's name and functionality. Lysozyme is capable of breaking bacterial cell walls. It causes lysis in bacteria, and therefore this very useful enzyme is excreted in every fluid of the human body. It's in our tears, our saliva, vaginal fluids, earwax, breast milk, sweat. You name it, it is there. In terms of containing damage and minimizing spread, a good example of this is the inflammatory response. This is triggered by histamine. There are many signals that say damage has occurred, but the predominant one is histamine. When this signal flare goes up, the end result is swelling, the flood of immune cells to the area. The inflammatory response is really a 5-step process. The first step being vasodilation. This occurs in response to that histamine flag going up. Vasodilation means that all the capillaries dilate, they get larger, and this increases blood flow to the area, hence why it quickly swells up and appears more red. But that increased blood flow leads to step number 2, chemotaxis, the chemical movement of a cell. Think taxi cab. So the immune cells hitch a ride on this extra blood flow to the area, thereby going exactly to where they need to be. Additionally, that blood will pour out of the wound, and in doing so can help clear the wound of any bacteria or debris that could otherwise be taken out. Margination is step 3. This is the process by which the immune cells adhere to the epithelium. They stop near the site of infection. Step 4 is diapedesis, when those immune cells actually leave the capillaries and blood vessels and enter the infected tissue. Then we hit step 5, and this is the ingestion or clearing or cleanup of the bacteria or other pathogen. which ultimately is phagocytosis, cell eating. Oftentimes, the immune cells that arrive to an inflamed site include natural killer cells. Their job is to clear any foreign molecule. And so what they do is they go through and look cell by cell to see, do you belong here? And what marks their trigger is the lack of an MHC class 1 flag. Every human cell in the body, an epithelial, a nerve cell, a muscle cell, you name it, has an MHC class 1 flag. It basically is an ID that says, I am yours. And natural killer cells float through the body and look for any cell missing this. Should they come across it, they ask no questions and immediately kill off the supposed invading cell. They do so through a dual enzyme action through the use of perforin and granzyme. Perforin creates a pore into the cell through which granzyme can pass through and go clear through to the inside and trigger apoptosis. Apoptosis is a special type of cell death wherein the cell virtually implodes, and it's a very safe death that releases no dangerous mechanisms, enzymes, or toxins.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The rath of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
Another way that we can clear or control or remove a pathogen is through the complement system. The complement system is a complex system that enlists over 40 different signal molecules and proteins, creating a waterfall or domino-like effect, which we refer to as a cascade. The end result is that these proteins can come together to increase further inflammatory zones, to flag for increased phagocytosis and cell eating, as well as forming membrane attack complexes, or MACs, which basically create pores in the foreign cell, resulting in death. So here's just an example diagram of the 40 different proteins that can come into being, complement protein 3, 3B, 3A, and how they can opsonize flag a bacterial species for phagocytosis, or trigger more histamine absorption for increased inflammation, or create MACs, which are very much like pores. Next up, we'll talk about the adaptive immune response. It's important to note that the adaptive system is actually a dual system made up of both T cells and B cells. Now, while they all originate in the bone marrow, they differentiate or mature separately in different environments. The B cells stay in the bone marrow and therefore get called B for bone cells, while the T cells mature in the thymus gland, hence the T. And because they evolved and matured in different areas, they have different specializations. So as we move forward, you will be expected to know the difference between the humoral and the cell-mediated responses. We'll begin with the humoral response. The humoral response involves the B cells. They operate on a self-not-self basis, very similar to the natural killer cells. They float through and they're looking for some sort of external pathogen, a bacteria just hanging out there in the open, or a parasitic worm sitting out there. They can recognize that as being foreign and will burp up antibodies to combat it. Know the difference between an antigen and an antibody. The suffix tells you where they come from. Antigen is like the pathogen. It refers to a carb or protein surface molecule that is foreign to the human body. The antibody comes from our bodies, and these are matched components that specifically bind to and neutralize the antigen. What we can see in the diagram here is a bacterial cell that's coated with triangles and mailbox shapes and squares. These are different antigens. And we can see that antibody A binds specifically to the triangle shape, while antibody B binds to that mailbox shape. The antibody is made up of 4 chains. The bottom portion, known as the constant region, and the variable portion. What I'm showing you here is how those areas correspond to gene segments in our DNA. And so as a result of different joining segments, we can give rise to close to 1 million different types of antibodies, meaning that at any point in time, you have roughly 1 million unique antibodies flowing through your bloodstream. Each antibody, though, is specific. It's able to recognize and react to only one antigen. Their variable regions, the tips of the Y, alter in shape, size, structure, charge, and so that is unique to them, and in theory, unique to a pathogen. Affinity refers to the strength of that attraction. Once they bind, do they hold steady indefinitely? And avidity refers to how much binding might take place, just one binding or multiple bindings together. Cross-reactivity refers to the ability to react to 2 antigens that might share the same epitope. Antibodies come in different forms, and as we discussed before, form dictates function. So antibodies G, D, and E, also known as immunoglobulins, G, D, and E, are monomer units. They function by themselves. Antibodies G, D, and E, also known as monomers G, D, and E, function by themselves as individual units. And as a lone wolf, they're most useful at fixing complement, triggering that cascade of 40 proteins. Antibody A, on the other hand, is the only antibody that exists as a dimer, 2 antibodies basically side by side. By having 4 arms, as opposed to 2, it is much more efficient at clumping and clearing, or agglutinating things, than its monomer counterparts. And antibody M is the only antibody that exists as a pentamer, a group of 5 antibodies together, which has even better agglutination potential. Once bound, antibodies will result in one of 4 actions. They can opsonize and flag something for phagocytotic disposal. They can cluster things or agglutinate them for a more rapid response or clearing response. They can neutralize it. If they bind and surround the entire cell or viral capsid, they can make it so that organism cannot bind to and enter or infect a neighboring cell. And lastly, they can be used to recruit other cells. They can trigger complement or stimulate T cells to come, which are involved in direct killing. The cell-mediated response, on the other hand, that are capable of targeting intracellular pathogens, transplant tissue, and cancer cells. They do this in two mechanisms. There are two populations of T cells. CD8 T cells are known as cytotoxic killer T cells, while CD4 T cells are known as helper T cells. The CD8 T cells are advanced natural killer cells. They work with MHC class I receptors. Remember that every cell has the MHC class I receptor. So what happens is the cell is constantly taking a little inventory of debris inside it and presenting those internal particles to the surface and saying to the T cells, hey, is this normal? In doing so, it acts like an X-ray machine, giving the T cell the inside scoop as to whether or not the cell is making only host proteins or whether it's been co-opted to make something a little more nefarious. Can you imagine how the Trojan War would have played out if the horse was not made of wood, but of glass? In contrast, the helper T cells bind to MHC class II receptors. These receptors are only occurring on macrophages, dendritic cells, and other phagocytotic cells. The idea here is that these specialized cleanup crews present external debris and basically act like news reporters saying, hey, did you see this? Is this still a thing? Should we continue to scout, search, and destroy? A good way to remember the pairings is to think in multiples of 8. 8 times 1 is 8, and 4 times 2 is 8. CD8 T cells pair with MHC class I receptors, while CD4 T cells bind with MHC class II receptors. The results of T cell stimulation is apoptosis. Again, they are essentially an evolved natural killer cell. The CD4 cells, however, will move on to activate other B cells and other T cells, and therefore that's why they get their name as helper T cells. These cytotoxic T cells also kill through perforin and granzyme, just like the natural killer cells. The benefit of the adaptive immune response is memory. Now, both the humoral and cell-mediated responses have memory. This results in more immune cells being present, already available, which means that the next time you come into contact with that pathogen, they're able to respond near immediately. And so ultimately, the human host has a shorter recovery. Vaccination is built on the concept of immunological memory, whereby early exposure to a pathogen could protect you from future disease. Historically, we came across this with respect to smallpox. During a time when smallpox was running rampantly through Europe, it became clear to Edward Jenner, the father of immunology, that milkmaids never seemed to get smallpox, despite it killing 25% of those infected. And so he went to the milkmaids and inquired about what they were doing, what their exposure was. And he noticed that they had smaller pustules on their hands, but instead of being red and itchy and smallpoxy, they were white and subtle. Turns out it was cowpox, which is not typically a human disease, but because these milkmaids were constantly with the cows, they had become susceptible to the disease. And he found that the early exposure to cowpox, or rather, he hypothesized that the early exposure to cowpox, being a close cousin of smallpox, would be able to protect them from future disease. And where not completely resistant, might result in a much less shorter or severe illness if smallpox were encountered in the future. If enough of the population is vaccinated, we get something known as herd immunity. The panel on the right demonstrates herd immunity. If most people get vaccinated against something, they get a gold star. And this means that should someone unvaccinated and infected with the disease, such as that man in red, come into the community, everyone there that he is surrounded by, everyone that he encounters, would not be susceptible to the infection and therefore not pass it on. And so despite there being a few blue individuals who are not sick and unvaccinated, only one actually gets ill. So this decreases the likelihood of something taking hold in a community. The panel on the left is what happens when we do not have herd immunity, when less people are vaccinated. Therefore, whoever that man in red associated with were susceptible and able to pass it on to those that they associate with in a rippling effect through the community. How much is required for herd immunity? It really depends upon the contagiousness of the disease. Something like measles needs a vaccination rate of 90% to be protective, whereas something like flu would be fine with only a 70% vaccination rate. Now, the immune system is great, but it's not infallible. Immunodeficiency describes a scenario in which there's less immune cells overall. And this can happen as you get older or due to changes in your health, your diet, or your own genetics. Part of immunodeficiency involves overstimulation. Perhaps your immune system doesn't know how to wind down. Perhaps the regulatory T cells that help moderate this response are not functioning correctly. Our immune system actually has a process known as tolerance. Tolerance, with respect to the immune system, involves the silencing of an immune response unless it's co-stimulated by another cell. The idea here, especially with T cells, which are capable of killing host cells, is to make sure that no one T cell is going rogue, that T cell has an override switch just in case it's the only one who sees a threat. Essentially, the immune system has to be regulated to prevent wasteful or unnecessary responses to harmless substances, and more importantly, to prevent it from attacking the host cells alone. Overstimulation of the immune system can lead to hypersensitivities down the line, commonly known as allergies. And if we train our immune system, we can stimulate tolerance. The concept of allergy shots is based upon eliciting tolerance, showing the immune cells that these other foreign substances are in fact harmless, and to silence reactions as you move forward. But if overstimulation does not lead to tolerance, the alternative is anaphylactic shock, which can be lethal. Another example of immune system malfunction is autoimmune disease, in which case your immune cells actively attack your body's own cells that are not infected by a pathogen. The cause for autoimmune disorders is largely unknown, although we do see a genetic and heritable component. And while our immune system is amazing for keeping us safe and healthy from pathogens in the world, sometimes it can complicate life-saving possibilities, such as transplantation. 50,000 people a year receive an organ transplant, while another 400,000 people are on wait lists for it. Among these transplants, 1 in 5, roughly, are rejected due to the immune system killing off the newly transplanted organ. This is why typing our individuals is so critical, because when we look at the recipient and the donors of an organ, we are trying to match both blood type and MHC class 1 tags. But as we mentioned before, due to exon shuffling in our genetics, the chances of an MHC class 1 tag being identical to someone else is roughly 1 in 20 million.